welcome back to Ultimate School. Today I am going to discuss with you about synthesis of thyroid hormones. As we know, the thyroid hormones are synthesized in the thyroid gland. And if you take thyroid is formed of follicular cells. Inside the thyroid follicle, there is colloid, and that is what is shown here. Now, for synthesis of thyroid hormone, we require two things. One is iodine, second is thyroglobulin. Let us look into the steps involved in production of thyroid hormone. From the blood, iodine enters the cell. Here what is shown is a single thyroid follicular cell. From the blood, iodine enters the thyroid follicle by an active transport, that is sodium-iodine symporter. The energy is utilized for the sodium-potassium pump, that is the 3-sodium-potassium pump energy is used, due to which 3 sodium is pushed out and 2 potassium goes inside. This produces a electrochemical gradient, and because of that sodium enters along with iodine also enters. This is the sodium-iodine symporter, and this is how iodine is trapped inside the cell. And this concentration can be 50 times. That is the usual thing that is seen in the thyroid follicle. That is 50 times more than that of the blood. It is concentrated. At times of need, this can be increased even up to 250 times. Hope this point is clear to you. Once the iodine enters the cell, what happens to the iodine? At the apical region, with the help of thyroperoxidase, hydrogen peroxide, the iodine is oxidized. So second step is oxidation of the iodine. And this oxidized iodine enters the colloid with the help of pendrin. So it passes into the colloid. So the next step is the thyroid, the thyroglobulin should be there. So let us look into how the thyroglobulin is synthesized. In the endoplasmic reticulum and later with the help of Golgi apparatus, the thyroglobulin is formed, and this reaches the colloid due to exocytosis. Here you can see the exocytosis, it enters the colloid. So now the two materials for the production of thyroid hormone is there. One is the thyroglobulin and second is the iodine. The iodination of the thyroglobulin is called organification. So here what is happening, in the thyroglobulin there are tyrosine residues are there. Tyrosine can attach 4 iodine molecules, and with the help of thyroid peroxidase, iodine is attached with tyrosine. When one iodine atom or one iodine attaches, it is monoiodotyrosine. When 2, it is called diiodotyrosine. One monoiodo combines with one diiodo to form the triiodotyrosine, that is the T3. And when 2 DIT combines together, it forms tetraiodotyrosine, that is the thyroxine. So now we have studied how the organification is occurring and how the T3 and T4 are formed. T4 is the major hormone that is released. Up to 93% is T4 and 7% is only T3. But the active form is T3. So this picture shows again a review of what has happened. Iodine is trapped, iodine is oxidized, it enters the colloid where monoiodotyrosine and diiodotyrosine are formed, which combine in various combinations forming the T3 and T4. And now what is happening? The hormone is preformed and stored in the colloid, and it enters the thyroid follicular cell again. It is via the pinocytosis. That is, it enters the follicular cell, where lysosomal enzymes act on, and what happens? Free T3 and free T4 are released and they enter the plasma, and the remaining diiodotyrosine and monoiodotyrosine deiodinated and reutilized for the same process again. Now we go in little more in the structure of the T3 and T4. 3, 5, 3-5- is the T4. 3, 5, 3- is the T3. That is, and there is another type called the reverse T3, where it is 3, 3-5-. So depending upon the position at which iodine is attached, we get the T3 and reverse T3. Reverse T3 is not active. So far we have discussed the iodine trap, oxidation, organification, endocytosis, proteolysis, and release of the thyroid hormone into the cells, into the plasma. In the plasma, this reaches in the blood, it reaches various organs, where it is peripherally converted into T3. That is, T4 is the major hormone, and that is converted into T3, which is active form in the periphery. So, hope you understood the production. In the next session, we will discuss on the hypothalamo-pituitary thyroid axis.